Hi, everyone. Welcome to the Going Organic Seminar Series. We're thrilled to have you with us today. Each month, we'll bring together researchers, growers, and anyone interested to explore and highlight the latest research and fresh ideas in organic agriculture, crop biofortification, and sustainability. We have a great session ahead. Dr. Lyndon Porter will be talking about integrated disease management in peas, chickpeas, dry beans, and lentils, a topic with valuable takeaways for breeders, agronomists, and producers alike. My name is Tristan Lawrence, and I'm filling in for Dr. Joe today. And joining me later for the Q&A session is Mark Dempsey, who will help guide your questions and discussion. We're excited to host today's session and glad you're here. Before we begin, I'd like to take a moment to acknowledge and thank our supporters, the USDA National Institute of Food and Agriculture, South Carolina Department of Agriculture, USDA Agriculture Research Service, Foodshot Global, and our collaborating institutions, colleagues, and students who contribute to this work. We deeply appreciate your support and partnership and contributions. Just a couple of quick notes before we get started. This session is being recorded, and we'll share the recording on Clemson's Pulse Breeding YouTube channel shortly after the webinar. For questions, please submit your questions anytime using the Q&A panel. We'll address as many questions as we can during the Q&A portion at the end. Feel free to use the chat to introduce yourself, share where you're joining from, or share any quick comments or resources. Dr. Porter's talk will run about 30 to 40 minutes, followed by 5 to 10 minutes of Q&A. All right, with that, let's meet our speaker. I'm excited to introduce today's speaker, Dr. Lyndon Porter. Dr. Porter is a research plant pathologist with the United States Department of Agriculture, where he has served for 21 years focusing on integrated pest management of bean, pea, chickpea, and lentil diseases. He has earned his doctorate in plant pathology from Washington State University and his master's in botany from Brigham Young University. He's originally from Provo, Utah, and now resides in West Richland, Washington, with his family. Outside of the lab, he's an avid outdoorsman. He enjoys hiking, fishing, camping, and just about anything that gets him out into nature. In collaborative research, Dr. Porter has helped develop a genetic marker for rapidly identifying pea lines resistant to pea and Asian mosaic virus. He's contributed to identifying 4 genes associated with Fusarium root rot resistance in peas, supporting the release of 5 pea lines with high levels of resistance that are being used by several private breeding companies. We're very glad to have him with us today. Dr. Porter, welcome. The floor is yours.
But today I'm just going to talk about Pythium root and seed rot, and also Fusarium root rot due to time. So this is a look at the different cultural practices. This is a look at the different integrated practices that that can be used to manage pulse crops, and especially organically. There's cultural practices. These cultural practices include things like planting date. They include planting depth. They also include seed spacing and row spacing, and also tillage practices. There's also different things that can be done with I'm going to talk about the importance of seed quality and seed treatments. I'm going to talk about biological and chemical applications that can be used, and the importance of plant nutrition in helping to manage different diseases and pathogens. And I'll also talk about, especially in organic production, how critical genetic resistance is when we're limited in the different things that we can apply due to growing organic crops. So this is, I'm going to show you, using a laser pointer here, and this is a chickpea field. And so the first thing that I want to talk about is just Pythium seed and seedling rot. And this is a picture from Weidong Chin, and this is a chickpea field. And I just wanted to point out Pythium seed rot can cause major stand problems. In organic production, when you have a stand issue, the number one culprit that you first want to look at is Pythium. Pythium across all of our crops, if you have poor stand, is the likely culprit to take a look at, especially in organic production. In this particular chickpea field, you see these bare patches that are all throughout. You see these low areas of stand counts where you see the soil a lot easier. And this is Pythium. This is a conventional field that was treated with metalaxyl, which has been used for years to manage Pythium. But there is metalaxyl resistant Pythium, especially in the Columbia Basin where I work with our organic green pea production, as they've transitioned from conventional growing conventionally to growing organically. They still can have major issues with metalaxyl resistant Pythium in their organic fields. And the problem with this is that the Pythium, metalaxyl resistant Pythium, is more aggressive than metalaxyl sensitive Pythium. And so and it continues even despite growing organically. The metalaxyl resistant Pythium persists in the soil, can be problematic for growers. So this is a look at this is a look at pea that's been infected by Pythium and has rotted in the soil. And when you dig this up, you can use a strainer, dig up the soil and wash that soil away, and you can get a look at what might be infecting your seed below the ground if you're having stand count issues or stand emergence issues. This is what Pythium seedling seed rot looks like. It's really an infected pea seed, looks caramel color, and the soil is impregnated into the seed, and you can't really scrub it free. And so this is this is what you might find if you're trying to find seed that's been infected by Pythium. You also this is what it looks like with chickpea. You get this brown discoloration, and you can see that a bunch of the shoots started to grow up on the chickpea here, but the Pythium pinched it off and stopped the growth and rotted the seed and the seedling that was developing. So it can be very problematic in our fields, especially organic chickpeas are extremely susceptible, the Kabuli type, to Pythium seed rot. This is a look at what Pythium seed and seedling rot can do. Over on the right-hand side here, you can see what happens with you can see infected seed that wasn't able to get up and out of the ground at all. You can also see the varying stages that Pythium of damage that Pythium can cause to a growing seedling. You see all of this pinching off that takes place of the secondary roots, and really overall either prevents it from getting out of the ground, or when the seedling does emerge, it's bigger and it's very weak, so it grows poorly. So this is the primary Pythium pathogen, probably across the entire United States and especially in our area, is Pythium ultimum. Pythium ultimum, that's the species of Pythium that's causing our major seed and seedling rot problems in our organic production, especially the seed rotting. So Pythium ultimum is a soil-borne pathogen. It's present. It's called a water mold, meaning that it's water-loving. In fact, it'll form what are called swimming zoospores that can follow a chemical trail of chemicals that are released by the roots or the seeds of plants, and they can follow that trail directly to where the seed or the roots are growing and infect them. This is a look. This is a look at a picture of oospores, which are the survival structures of Pythium ultimum, and they can survive in the soil, they say, for up to 10 years. So they can, even though you may not plant a susceptible crop, they can survive in the soil for extended periods of time and still cause problems. But one problem with Pythium is it is a generalist, and just about every single crop that we plant is susceptible to Pythium, especially Pythium ultimum. As I said, Pythium ultimum is ubiquitous. It's This is the primary Pythium pathogen that we're really finding across the United States based on the literature and based on the research that we've done here in Washington and Idaho and Oregon. And they're also, Pythium is a rapid colonizer. So it's able to move through the soil extremely quick. In fact, from all of the different fungi that I studied that are pathogenic fungi, it is the fastest growing fungi that there is, and can move through the soil very quickly. So it can locate the seed or root system in the soil very quickly, and can grow inches in a day. So, as I said, conventionally, Pythium ultimum has been controlled using metalaxyl or metalaxyl-Lim fungicides. But in organic production, we don't have this. We don't have that luxury. So I'll talk a little bit later about what we can use to manage Pythium. So, I've done across some organic pea fields in Washington. We took a look and we surveyed the different Pythium species that were causing problems in these organic fields. And this is a list of the different Pythium species that we found. We plated these on a Pythium selective medium called PARP, and then we were able to isolate the different Pythium from that medium, and determine what Pythium species we have in the soil. Those species that are circled are pathogenic Pythium species to pea and to other pulses. And you can see that Pythium ultimum, live cultures of Pythium ultimum were found in about 85% of the fields that we surveyed. But you can see the other Pythium species circled here that are pathogenic that were also present. So, but this Pythium ultimum is the major Pythium, and it's aggressive and it's very fast growing, and causes major issues in our organic production. Not only in pulses or peas, green peas, but in all organic produce. So this is a look at, so in these same 37 organic pea fields, we isolated, we took a look at the DNA concentration of Pythium ultimum. We had what were called selective DNA primers for Pythium ultimum, and we were able to find it in 100% of our pea fields. Of the 37 pea fields, we were able to detect DNA that was specific to Pythium ultimum. Now, one thing I want to point out, across these 37 organic pea fields that we took a look at, you can see that the level of DNA of this pathogen varies greatly. But the one thing that you can do as a proactive organic grower, realizing how impactful Pythium ultimum is on your stand counts, if you're able to select your fields that you're going into to plant your highly susceptible organic crops to Pythium, such as chickpeas or Kabuli chickpeas, then you can take a look at your concentration of DNA of Pythium ultimum in those soils, and be able to select a soil that has much lower levels, and give you the greater opportunity to have excellent stand counts in those fields. So this is something that you can do as a proactive organic grower, is to know these levels in your fields. This is a look at Pythium soil counts in two different organic fields. These are organic green pea fields. One in Moses Lake, and these are the different replicated areas within that field. And then another area in Patterson, Washington, in replicated fields. And this is the number of Pythium colonies that we're growing on agar plates per gram of soil. And this is the number of metalaxyl-resistant Pythium per gram of soil. So even though these were organic fields, these metalaxyl-resistant Pythium that are more aggressive still persisted in the organic production, and were problematic because of their aggressiveness for organic growers. But what I want to point out, anything over like 10 10 propagules per gram of soil is still a pretty good level of pythium. But you can see we have in this particular organic field, we had over all of the replications had over 440 propagules per gram of soil. But in Patterson, they were a lot lower. So your different organic fields can vary in the level of pythium that's present in that soil. And if you're having poor stand counts, it's important to know what your pythium counts are, um, to be able to know what risk you're taking by planting organic produce into those fields. So as far as managing pythium, I want to talk about some different things that you can do to manage pythium in your organic production fields. I'm going to talk about seed quality, soil moisture, soil temperature, how you can adjust your planting depth, rain events or irrigation events. I'm going to talk about seed treatments and also talk about something called seed priming. And these are some different things that you can do under organic production practices to help to mitigate and manage pythium. So the first thing that you want to do as an organic grower is you want to make sure that you are planting into your soil the highest quality seed that you can find. And it's critical that you take a look at your seed and look for abnormalities. Some of the abnormalities, this is an example of what pea seed-borne mosaic virus can look like on pea seed. This pea seed-borne mosaic virus, first of all, viruses and split seed or seed that was harvested and the harvester kind of roughed up that seed and kind of interrupted the integrity of the seed coat, and causing split seed coats. So viruses can cause this. Poor water events that hydrate your seed before harvest can cause poor seed quality. This is a particular problem with manganese deficiency in your field for peas, and it causes this internal discoloration called marsh spot. And this is a look at some damage that can cause during pea seed formation when you're with high temperatures and other things that can cause something called hollow heart. Now, all of these things can impact the vigor of your seed. And when you impact the vigor of your seed, you impact the speed with which it emerges out of the soil. And the faster it can get up and out of the soil, the less of a problem pythium seed rot becomes. Because when it is able to start to photosynthesize, then pythium becomes less of a problem. The other things that poor seed quality does is seed that has poor seed quality tends to release higher levels of sugars and amino acids and other things when you plant that seed in the soil. And those sugars and amino acids that are released in the soil trigger the response of the pathogens in that soil to a higher degree, and they're able to find the seed and infect the seed or the root system. And so that's why you want to plant the highest integrity, the highest quality seed you can find. So this is just some look at some other seed quality issues in chickpea. You can hydrate up some of this seed and cut it open and take a look at the inside and take a look at internal defects. And so this is what a healthy on the left. These are healthy chickpeas, and on the right, these are some of these internal defects that we can find that can dramatically impact the vigor of your seed. So there are seed-borne pathogens, not just viruses, that can also reduce your seed quality. There was a grower that was growing organic peas, and he was concerned about his poor emergence, and he sent me some of his seed, and I just took a look at his seed. And within that seed, this is some of the seed that I gathered out of there. And you can see that it's not normal looking. It's got brown splotches all over it. And when I plated this out on an agar medium, you can see the fungus that is growing out of it, and this was Ascochyta blight. But there's other fungi, such as Alternaria, Aspergillus, Ascochyta, and Fusarium, that can result in really poor seed quality and impact the vigor of your seed. So you want to inspect your seed personally. Take a look at that seed before it's treated with any sort of colorant, if you're putting a seed treatment on there, to make sure you understand just visually how what the quality of your seed looks like. And you want to plant high quality seed. So this is a way that you can determine seed quality visually, but you also in organic green pea production, um, organic growers use what's called they look at what's called electrical conductivity to try to determine the vigor of their seed, and they can classify the vigor of their seed based on what's called electrical conductivity of water. When a given amount of pea seed is soaked in the water at a specific temperature for a given amount of time, then you're able to use what's called an electroconductivity meter and measure the electrical conductivity of that water. The lower the electrical conductivity, measured in what's called a unit called Siemens, the higher the vigor of the seed is. And so the higher your electrical conductivity, the poorer or the lower the vigor of your seed is. And what is happening is the integrity of your seed and the internal integrity of that seed, if it's poor, they'll release a lot more amino acids, sugar, organic acid, and other compounds into the water, which increases the electrical conductivity, its ability to conduct electricity. So a lot of our organic pea growers will take different seed lots, and prior to planting, they'll take a look at the electrical conductivity of that seed. And they're looking for seed lots that have very low electrical conductivity. For example, this particular cultivar, and seed lot 1 had a very low electrical conductivity, and which gives it a high vigor ranking, and it also had good germination. And so as an organic grower looking for the best seed quality of seed that you can, um, in peas, you can do this test to be able to determine your seed quality. And you want to go low electrical conductivity and high germination. And that's the seed you want to plant and going to give you your best results. And it's going to get up and out of the soil faster and give Pythium less of a time to find and rot the seed and rot the seedling before it gets up and out of the ground. So this is this is a screening, nice photo from Dr. Weidong Chen at a nursery that he has, where he'll screen for different things like Pythium and Phantomyces root rot that can cause problems on peas and lentils. I think pictured here are chickpeas. But what I want to when we're trying to um, test different seed treatments for resistance against Pythium, or to help fight against Pythium, we plant into saturated soils, and or we saturate soils immediately after planting. And so saturated soils, you don't want to plant into saturated soils in organic or conventional. You want to plant into soil that has adequate moisture, that allows you to go the longest period of time before you need an irrigation or a rain event to continue the growth of that seed. And so you don't want to plant and then water if you have irrigation capabilities, or you don't want to plant and have a heavy rain event that happens. Those are all going to favor Pythium seed rot in your fields. You also want to plant when your soil temperatures are least above 50 Fahrenheit, and this once again improves the speed at which your plant germinates and emerges from the soil, and will reduce problems with Pythium. And then once again, you just want to recognize that rain events or irrigation events immediately or soon after planting will favor Pythium seed rot. So you want to, once again, we want to plant into adequate moisture and wait as long as possible until emergence. You want to monitor your soil moisture levels, but you want to wait as long as possible before you have another irrigation event, if possible, if you control irrigation. So planting depth is also important to avoid problems with Pythium. You want to plant as shallow as you can. Some of our organic pea growers will plant at a half inch. Sometimes that's difficult to avoid erosion or to get sufficient soil cover. But the principle is planting as shallow as you can get away with will help that plant get up and out before Pythium can find it and infect the seed or infect the seedling. And once it gets out and starts to photosynthesize, Pythium, seedling, and seed rot become less of an issue. So, what can you do besides these cultivation practices and planting practices? Are there organic seed treatments that can be used? And so this was a trial that I conducted, taking a look at the ability of different organic pea seed treatments. And what you see, this is a water controlled. The seed was only treated with water, and you can see the pore stand count here. Only 14.9% of emergence. And here we have Apron Maxim. That's a classical, non-organic, conventional treatment, but we needed it in there in this particular field to see what the Pythium pressure was like when we have something that works well to control Pythium. This Nordox is cuprous oxide, which we were taking a look at as a potential seed treatment, organic seed treatment for peas and other things. And this is a product called Ultim ST from Germain Seed Technology that uses copper hydroxide. And this is a OMRI certified organic seed treatment for now for peas and chickpeas and peanuts and corn. But the exciting thing was that we found that this Ultim ST with copper hydroxide, we got 72.8% emergence compared to planting just non-treated seed. And this particular product, it's not a biological, obviously it's copper hydroxide. It's very reliable as far as its efficacy in managing Pythium as an organic seed treatment, and it's an exciting new product. You can This is when we took a look at the yields compared to the water treated. The Apron Maxim, that conventional treatment, was 111% increase in yield. The Nordox was 40.6% increase in yield compared to the water control. And this Ultim ST is the best product that I've found in all the different biological and chemical products that are organic products to manage Pythium seed rot. So some other things that you can do is seed priming to try to avoid problems with Pythium seed rot. Seed priming is where you soak your seed in sterile water prior to planting it. So pea seed you can soak for example 16 hours. Different chickpeas and beans you can't soak them that long to prime them. But you soak the seed up, you hydrate them up, and then you can dry them for 10 hours and then plant. You can also apply some sort of desiccant to it to dry the water up and then plant as well. And so we found that there's faster emergence, and it also can reduce these leachates that are released from the seed into the soil that attract Pythium and Fusarium and other soil-borne pathogens, and you can reduce rot due to those pathogens by priming as well. So we took a look. We did two organic pea seed treatments in Boardman and Soap Lake, Washington. This is a look at this organic product that we were looking at. That was this cuprous oxide. But this was a look at primed seed, just priming them for 16 hours and putting them out. And these are two different seasons that we conducted the trial, or the two different locations. And you can see that this priming just by itself can be very effective in helping the seed to get up and out of the ground and avoid problems associated with Pythium compared to some of these other biologicals, and no seed treatment at all. Nutrition is also critically important for good root health, to help your plants avoid problems with root rot pathogens, and or to help them to better overcome root rot pathogens that may develop, that due to a vigorous plant is not as severe. And in our soils here, especially in organic production soils, we have problems with low pHs. Anything that's so you want plants that are you want soil that's above a 5.5 pH when you're planting peas, for example. We also have problems with low sulfur, low zinc, and low boron. And so you want to make sure that you test your soil. This was with the the different soil tests that you can have. But if you have low levels, then you want to take a look at different organic applications that you can make associated with these different elements, nutrients, sulfur, zinc, and boron, to elevate those, and you'll improve your overall root health as well, and plant health. So as far as management of Pythium seed and seedling rot, there are you want to plant quality seed. Take a look at that seed, and also like with fresh green pea, as I talked about, you can actually take a look at the vigor based on electrical conductivity. You want to do those things that will allow rapid emergence from the soil, that will help avoid Pythium rotting your seed. That means planting into those warmer soils, shallow planting, as shallow as you can get away with, and adequate soil moisture, and then that you plant into, and then don't add, if you can irrigate, or you don't want to irrigate immediately after planting. If you have adequate soil moisture, delay that until emergence if possible. You want to, once you get that plant photosynthesizing, Pythium is less of an issue. So there's this new organic product, ProBio Ultim ST, by Germain Seed Technology. That's something that you should test and see if it works for you. It's the best product that I've seen to manage Pythium organically. And if you are going to use biological seed treatments, of the treatments that I've tested, it seems that those that are of a Bacillus subtilis type seed treatment have worked the best for those that I've tested over the years. But there's always new products coming out that need to be tested. So now I'm going to turn to Fusarium root rot. Fusarium root rot is caused by Fusarium solani, or Fusarium avinaceum, or a whole bunch of other Fusarium species. In peas, they'll cause this black seed rot and blackening of your seed, right around of your root, right around the seed attachment zone. So you can take a look at these different pictures of the black that it can cause. This is what it looks like in peas. You also with Fusarium avinaceum on lentil, it actually causes a rustic looking infection. And in chickpeas, different species of Fusarium will blacken that root system, and that black root system is a indicator that you may have Fusarium root rot going on there. So one way to manage Fusarium and also Pythium is to avoid soil compaction. Soil compaction is a major problem and can flare these diseases and problems with these diseases in your field. Why? Soil compaction reduces or slows plant emergence. So the seed is in the soil for a longer period of time until it emerges and gives Pythium and Fusarium a longer period of time to infect it. Before it emerges, it restricts the root development and growth. It lowers the oxygen level in the root zone, which can stress the plant out. It restricts water percolation, and for Pythium, it's a water mold, so it loves saturated soils, and it is able to better infect your root systems. But so these types of conditions, though, favor fusarium and pythium root rot and other soil diseases. So if you're doing constant practices such as packing your soil when the soil is wet, too wet, that increases the compaction layer. Then if you're continuing these types of practices that favor soil compaction, then you're also favoring the buildup of these pathogens in your soil over time, and you're going to have more and more problems with them as you continue with poor production practices that favor compaction. So one of the best ways organically to manage fusarium root rots is through genetic resistance. And so a lot of what I do is I screen a lot of this is green dry peas, green yellow peas, Austrian winter peas, green fresh peas, yellow winter peas. And so I screened these different cultivars for resistance to Fusarium solani and Fusarium avinaceum, which are the primary Fusarium root rots. So you want to figure out which of these, especially for organic production, where you can't really apply anything to manage Fusarium as a seed treatment, you want to be able to use, you want to be able to plant the most resistant lines in the ground that you can when you have that choice to make. So this is just a look at the resistance on a scale from 0 to 6, with 6 being highly susceptible. These are a look at all these different pea lines, and you can see the highly susceptible and the resistant lines, and what their different cultivar is for these. These blue ones are Austrian winter peas, which are the most resistant to Fusarium solani. But as an organic grower, you want to know your genetic resistance of your different lines that you can plant against the major pathogens that are problems in your field. This is a look at it, pea resistance to Fusarium avinaceum, which is more aggressive than Fusarium solani. And you can see that Melrose, these Austrian winter peas, were the most resistant. But you can see which lines are the most resistant. There's not a whole lot of resistance in pea lines to this Fusarium avinaceum, but you want to plant your most resistant lines that you have to give you your best opportunity for good yields. This is just an example. This is a particular cultivar that's not inoculated with Fusarium. You can see how healthy the root systems are and the plant and the shoot growth. This is that same variety inoculated at the same time with Fusarium avinaceum, and you can see how devastated it is. And this is when that same variety is inoculated with Fusarium solani. So you want to be able to figure out and have researchers take a look at the levels of resistance of the different cultivars you can choose to plant in your field, and plant cultivars with the greatest genetic resistance to the problems, to those pathogens that are problems in your field. So I screened over 444 lines, pea lines, for resistance to Fusarium avinaceum, and I was able to find a number of different lines from pea lines from pea seed from all over the world. And then these had extremely low on a 0 to 6, with 6 being a high, very low, below 1 disease severity. And we're able to start using these in our breeding programs to try to develop resistant lines to Fusarium. And so that's the value of identifying genetic resistance. So these are different lines that we were screening in a Fusarium root rot nursery to find which lines are best for conventional peas as well as organic pea production. And you can see these two lines that survive extremely well in this field that has Fusarium root rot and Fusarium wilt. And you can see that we can select these lines, and then these are the lines that organic producers can use or conventional growers can use for pea production. So for Fusarium management, you want to avoid compacting soil, and you want to know how susceptible your different cultivars are that you're planting, and also work with breeders to develop more genetically resistant lines. And in conclusion, with my talk here, You want to start with the best quality seed available to manage Pythium and Fusarium root rot. Pay attention to soil moisture levels and temperatures at planting and forecasted rain events. And you want to select planting dates that will favor adequate soil and moisture, but not saturated soils, which favor Pythium. Don't plant seed deeper than necessary for Pythium to avoid Pythium problems. This Ultima ST is actually kind of a groundbreaking organic product, seed treatment to manage Pythium. Priming seed can be used, but it's a little bit more time consuming and can be a little more tricky. But it does work to improve emergence. Um, and you also want to identify the major economic pathogen in your growing area and screen crop germplasm for resistance to this pathogen, or the cultivars that you're going to use for resistance to these pathogens. And that will help improve your yields and your productivity and the risks that you have. And with that, I'll take any questions that anybody might have. This is a look at a beautiful Palouse Falls that's near where I live. And in the corner here, you'll actually see my buddy rappelling. We rappelled down the face of this wall, and it was pretty crazy. I'll take any questions that anybody might have.
Thank you so much, Dr. Porter, for your excellent presentation. That was great. So we're going to go ahead and move into our Q&A session. So if you haven't already, go ahead and enter questions to the Q&A panel, and we'll address as many as we can with the remaining time. I'm going to hand things over to Mark Dempsey to start us off with the first question. Thank you, Mark.
Yeah, sure. Thank you, Dr. Porter, for the excellent presentation. That was informative, and hopefully our organic pulse growers out there are tuned in or able to tune in later to soak up some of this good information. I'd like to start off with a broad few questions. So it sounds like you presented a lot of multiple management strategies that might be necessary in combination for production success. Is your take on that those multiple methods, that they should be implemented sort of together as a suite of practices, or could a grower focus on just one or two and hope for success?
A grower can, to the level that they can start with one and then they can build. And as long as you're aware, you've got the knowledge of the different tools that you can use, such as planting date, planting depth, tillage practices. I didn't really get into tillage practices, but different things that reduce soil compaction. Implementing all of those, a progressive grower that's trying to minimize his risks will try to implement as many of these practices as possible, and it'll result in a higher yields.
Sure. Yeah. And so to pivot a little bit about or maybe to be a little bit more inclusive of other pathogens that we didn't talk about in detail today, acknowledging that sometimes, or maybe often, root rots are a complex of pathogens that might be affecting soil. Does the pathogen identity, or like various identities within that complex, make a big difference in what or and how we might manage? Or are those core management practices applicable to a root rot complex?
Yeah. So the interesting thing in organic, the reason why I focused in on Pythium is if you don't get your crop up and out of the ground to begin with, you don't have to worry about the root rots, the other root rots that come. So the first thing you want to do is make sure you get your good stand count by good Pythium management strategies. And then after you're successful at getting them up, they're really for organic root rot management. It's at the very it's planting that quality seed. It's all of this upfront, very early management stuff that determines your success. And then because conventionally there's more things that you can spray and you can apply to try to manage Fusarium root rot after the fact. But with organics, you want to plant the best with it's really all of this upfront thing I talked about, the best genetics and then all of these other cultural practices that are really, and then the only other thing that can be the biggest difference is your irrigation management. If you are able to, if you're irrigating, if you're not irrigating and you just have to trust in Mother Nature, then that's a different thing. But you realize rain events and irrigation events after planting are going to be the major, and you just want to maintain adequate soil instead of super saturated soils, and that's probably your best options organically.
Yeah, sure. That makes sense. Um, so you had mentioned that Pythium, or that most crops host Pythium or are affected by Pythium. Are there non-host crops for Fusarium root rot?
Uh, all of our pulses are susceptible to the fusariums. There's some excellent work done out of Montana State University with the pulses that showed that fusariums cross infected. So as far as pulse production, and even fusarium and pythium are very generalists. Um, Aphanomyces is less of a generalist. Like for example, chickpeas are extremely tolerant or resistant, and lentils and peas are not. But our fusariums and our pythiums are very generalists across not only our pulses but other crops in rotation with pulses.
Yeah, and is it still advisable that longer rotations might improve those root rot outcomes?
Yeah, absolutely. So that is an important thing that I um, I talked about pathogen loads or levels that by compaction, but also crop rotation is critical, and to a healthy crop rotation where you're not planting pulses within the 3 or greater years out is highly recommended for sure. Crop rotation is a big deal.
Yeah, definitely. And so I'm curious. One of the seemed like a very good recommendation would be to shoot for warmer soils. And in spring planted pulse crops, does that simply mean delaying planting?
It can yeah. I mean, it does. It means delaying planting until those temperatures are where you need them to be. Growers recognize how long at different growers, depending on their acreage that they have to plant, and they recognize when they have to get in to be able to get the acreage and the water that they have if they're on dry land. And so but to the level that they can delay to have those higher temperatures, it's going above 50 Fahrenheit, and even for chickpeas into the 55 degrees Fahrenheit. The growers in the Palouse area they'll plant peas, then lentils, then chickpeas last. And the reason for that is the kind of almost based on a lot of it is based on that chickpeas are so susceptible that you want to plant them into the warmest soil so they get up and out the fastest. And your lentils and peas are more resistant to pythium than chickpea, a Kabuli chickpea.
Yeah, sure. And if I were to go out and want to measure soil temperature, what depth, out of curiosity, am I measuring to? Just the surface 2 inches or surface 6 inches?
Yeah, I think your main focus is just in where that initial seed is going to germinate, and then that the initial root zone that it'll occupy. But the most important thing is that where that seed is planted, and the depth of planting.
Yeah, absolutely. That sounds good. So to pivot a little bit and to go back to some of that quantification of Pythium that you'd mentioned, do you have a recommendation for early detection tests for Pythium or Fusarium or other pathogens?
Yeah, so there are different potting soil techniques that growers will use to try to determine. It's a labor intensive way to do it, but growers will take soil pre-plant and enough time to where they can um, plant seed into that soil and see how well it emerges. And that's a classic, and especially organic growers, do that, and they have an idea of just the level of pythium that may be in that soil, and it gives them confidence or no confidence whether or not there's going to be significant risks for their organic production when they planting into that soil. So yes, they will do that, but it is labor intensive. Um, but they can do small tests, and those small tests can even give you a decent idea of how much pythium you may have in your soil that might be causing seed rot.
Yeah, and and if it doesn't look good in the test, their options are to risk it and implement as many of those management practices we discussed,
Yep.
or avoid the risk and switch the crop or something like that.
They can switch the crop if they're able to switch fields because they have the options. They could switch to a field that had good where the soil had good emergence based on those tests. And so those are and they can also put their highest risk, highest pythium risk um, crop in a low pythium area versus higher area, and they can play around with that as well.
Sure. Yeah, that makes sense. Um, I'm curious about an additional technique that we didn't cover today that I've heard other folks talk about, either growers or researchers, and that's preceding a pulse crop with a mustard cover crop. Do you have any feelings about that practice, whether it might be worth the effort to have um, some sort of suppression from a mustard cover crop?
Yeah, the glucosinolates are the reason that they're planting mustard is that the isothiocyanates are what they're trying that are released from the mustard when the mustard is incorporated at a young, as that breaks down, it can release these isothiocyanates that are like a natural fumigant, and their their success with that, I think there's mixed results with that. And so I'm and so I guess I'm not prepared to say to what level that's a good practice or not, to tell you the truth. But one thing that it does do is it's a way for growers to increase their organic matter and try to increase the health of the soil as well. And so there may be some additional benefits there besides the fumigation effect from the mustards.
Sure. Yeah, that makes sense. So yeah, potentially helpful, but not reliable.
I think so. Yeah.
Yes. Yeah, sure. Okay. Well, it looks like our Q&A, we're done with any Q&A questions, and I'm gonna double check chat real quick. Give me 1 minute to read through a couple of questions here. So, Ashok Sa asks or mentions, last year I noticed an infestation of Pythian rot on transplanted watermelon after 10 to 15 days after transplant. Any suggestions to control during that phase of watermelon production? I understand we've switched crops suddenly. Um, but
So
yep,
watermelon transplants that the root systems went down hard because they were impacted by Pythium. Yeah. So there a couple things that I'd pay attention to. One is that I would make sure that during the transplant process, you're not damaging the root system. That and I don't know exactly what you're doing, but you don't want to damage the root system during the transplant. The other thing is making sure planting into adequate soil, but not saturated soils, and then not. You want to plant into adequate soil, and then let that. You want to then let that root system start to heal if there's damage from doing the transplanting, and you don't want to saturate up that soil really to a huge level because that's just gonna flare and get Pythium a greater opportunity. So those are maybe two things that I make sure I'm not damaging the root system during the transplant process, and then not planting into super saturated, oversaturated soils that would favor Pythium.
great. Yeah, hopefully that's helpful to Ashok. Yeah, they're doing a pivot to a different type of crop, but he's a PhD student at Clemson, and hopefully get some benefit out of this. So, That is actually the end of our Q&A, and I realize we're coming up on a full hour here. So I want to thank you again, Dr. Porter, for the really helpful presentation and Q&A, and pass it back to Tristan real quick for what's next.
Yeah, thank you, Mark. On behalf of the Going Organic team, I just wanted to thank everyone and Dr. Porter for attending and sharing your knowledge with us. Our next webinar is coming up on April 15th, and it's featuring Dr. Audrey Khalil, who's a pathologist, agronomist, and outreach coordinator at Horizon Resources Cooperative. She'll be joining and presenting on agronomic management of disease in pulse crops. We hope you join us for another excellent speaker on diseases. And
thanks so much to everybody.
Yep. Thank you so much. We'll post today's recording on our YouTube channel. And once again, thank you. And we hope to see you next month. Thank you, Dr. Porter.
You bet. Great to be with you.